Morning, everyone. Thanks for the invitation and for giving me this opportunity to introduce you recent findings in the parent of origin effects, dependent methylome, and the rate of aging in humans. The inspiration of this study should contribute to Barker's developmental origin of health and disease theory, which hypothesizes that some of our late-onset diseases, such as metabolic disorders, psychiatric disorders, and cancers, can be profoundly influenced by our early life experiences, such as prenatal exposures. Since aging is a fundamental cause of many late-onset diseases, it's reasonable to hypothesize that aging can be influenced by early effects as well. But in order to understand that, we need to work out what is the molecular mechanism that connect the early effects and the aging in later life. Among all possible mechanisms, I think epigenome can be quite relevant because during our early development, we experience intensive epigenetic reprogramming. So any external environmental exposures may influence the establishment of epigenetic status. And if that part of the epigenome further influences the adulthood health and aging, that will connect the early effects and later life aging. So now our question is which part of the epigenome is vulnerable to those early effects, and if that part of the epigenome can influence the aging in adulthood. So one of the candidate regions that came to our mind is the region influenced by the parent of origin effect. You can just tell from the name that this type of effect is established early in the life. Among all causes of POE, genomic imprinting is the most well-studied one. It's a phenomenon that was detected in a small part of the genome where only one of the two copies can be expressed. It's either maternally expressed or paternally expressed. This is in contrast to the biallelic expressed of both copies, which apply to the majority of the genome. So now, imagine that you're looking at a MQTL type regulation where a SNP genotype is associated with the methylation level of a nearby CpG site. If both alleles can be expressed, then we expect to observe an additive regulation mode. However, if the region is influenced by the POE, then it is not the dosage matters, but also the parent of origin of the specific allele also matters. So in this case, only when the A1 allele is from the paternal chromosome, it can influence the methylation levels. So one of the signs of POE is the unequal methylation levels between the two heterozygous groups. So the reason that we think the POE can be quite relevant when to explore the association between early effect and the aging in later life is because the imprinting itself is established during the epigenetic reprogramming in development stage. Specifically, there are global waves of demethylation and remethylation in the majority of the epigenome. So this will remove the parent of origin information, which should be traced back to the sperm or the oocytes. However, a small part of the epigenome managed to escape that process so that the parent of origin information can be remade. Those regions are indeed the imprinted regions. So you could imagine that if during this critical period of time, the organisms are exposed to environmental disruptions, then the POE process tend to be also influenced. So there are already some reports suggested that the baby who were born from the ART, which is assisted reproductive technology, they tend to have a higher risk to develop imprinting disorders. Because the ART procedure involves the gamete freezing, embryo culture and freezing and transfer, which all of them might influence the establishment of POE. So here, we propose a hypothesis that the POE-influenced methylome can be associated with the adulthood aging in the humans. But in order to test that, we need to first work out where the POE-influenced methylome are located. So how can we detect the POE at the population level? The answer is quite straightforward, actually. From the textbooks, we have learned that for a trait that is only influenced by the additive genetic effect, we will expect to see that for individual pairs who share the similar amount of the DNA, they tend to have also similar phenotypic similarities. So full sibling pairs will have similar phenotypic similarities as those parent and offspring pairs. However, also in the textbooks, there is a small part describing a non-additive genetic effects, parent of origin effects. It suggested that if it is the POE influence the phenotype, then we would expect to see the broken of such pattern. So if you want to detect the POE at methylation level, then you could treat individual methylation site as phenotype. And if you observed an imbalanced methylation similarity within nuclear family members of the same genetic similarity, then that means there is a POE on the CpG side. So in our 2019 publication, we used a virus component framework to identify CpG sites that display such imbalanced methylation similarity pattern. For example, for some of the CpG sites, we observed the increased father-offspring similarity as compared to the mother-offspring similarity. For some others, we observed the increased mother-offspring similarity as compared to the father-offspring similarities. So the data that we used to detect the POE-influenced methylation sites is Generation Scotland, which is a family and population-based cohort of adult participants from over 7,000 families. This cohort has rich information about the healthy-related phenotypes and exposures. And importantly, they also have genotype and DNA methylation information available. By the time that we performed the initial study to detect the POE at methylation level, there were around 5,000 participants have their DNA methylation data available. This allowed us to identify around 1,000 CpG sites for the POE as they displayed the imbalanced methylation similarity within nuclear family members. For around 60% of those CpG sites, we also detected the POE MQTL SNP because those SNP, they influence the methylation levels in a parent of origin way. So this exactly satisfy the hypothesis of genomic imprinting, which suggested that one of the two copies is expressed. And indeed, when we perform enrichment analysis for this part of the CpG sites, they are strongly enriched in the no-imprinted region. So we call this subset of POE CpGs as typical class. For the rest of the POE CpGs, we didn't detect any POE MQTL for them, and they are not enriched in no-imprinted regions. We further ruled out the influence from the dominance genetic effect. So we think that the imbalanced methylation similarity detected in those CpG sites are most likely to be introduced by the early familial environments. So we call those CpGs as a typical class. Now we have the information about the POE-influenced methylome. We can next testify our hypothesis of the association between the POE-influenced methylome and aging at adulthood in humans. By the time that we perform this as part of the analysis, the Generation Scotland has already produced another wave of DNA methylation data for around 4,000 individuals. This allowed us to have a discovery and replication setting in our association analysis. We collected more than 100 phenotypes that describe people's early life exposures, adult life exposures, disease and health conditions, and of course, aging status. The reason that we collect the early life exposure is because we want to confirm our hypothesis that the POE-influenced methylome are indeed sensitive to early effects. The reason that we use disease and health phenotypes is because aging is a fundamental cause of many late-onset diseases. So if the POE-CpGs are associated with aging, then they should also be associated with health phenotypes. In terms of the POE-influenced methylome part, we will use those as identified from our previous study, as I just introduced. We will perform separate analysis for the typical class and atypical class of POE-CpGs because, as I just introduced, they are likely to have different POE mechanisms. Now we have the phenotype and the POE-influenced methylome. We could perform the phenome-wide association analysis at both the single CpG and methylation network level. In both stages of analysis, we accounted for factors such as family structures, batch effect, age, smoking, gender, and cell proportion. So now I want to spend a few more minutes to introduce our aging phenotypes. We have 4 aging phenotypes in total, 2 of which describes accelerated methylation clock. I'm sure that some of you have heard about the epigenetic clock theory, which means that you could use a linear combination of methylation levels of a number of age or longevity associated CpGs to create an estimator of the internal aging status. Once you have that, you could draw a regression line with the chronological age, so that you can imagine that if you project every individual in your population into the figure, most of them will be distributed nearby the regression line. However, there are some of the people, they deviate from the regression line more than others. They either have an advanced age, which we call them as the positive epigenetic age acceleration, or they have a slowdown aging, which we call them as the negative epigenetic acceleration. So you could imagine that once you could calculate this age acceleration, then you have a measurement about the aging status in your population. In our sample, we calculated 2 acceleration phenotypes based on 2 published epigenetic clock. The first clock is the pheno-age clock, which is a DNA methylation estimator of a weighted average of 10 longevity-related clinical characteristics. So once we get this clock, we just use age to adjust it from the pheno-age clock. That gives us the acceleration phenotype. And the second clock is based on a DNA methylation-based estimator of telomere length. And once we get this, we use a regression to adjust the age from the predicted telomere length to get the telomere length acceleration phenotype. As we know that for the telomere length, a longer length represents a good health in general. So here we think that the higher acceleration represents a good health conditions. However, the pheno-age acceleration, the higher acceleration represents the poor health conditions, the more advanced aging status. So when we perform the phenotypic correlation analysis, you can see that the 2 phenotypes are negatively correlated. In addition, we also include the maternal lifespan and the paternal lifespan as another 2 aging phenotypes. So now I could share with you what we find. From the single CpG-based association tests, we observed widespread replicated associations of POE-CpGs with multiple phenotypes. Here, I only listed the phenotypes that are associated with at least 1 POE-CpG. The length of the bar represents the average significance levels of that phenotype. As you can see, the most significantly associated phenotype is smoking, which is not surprising given the global effect the smoking has on the entire epigenome. But the second most associated phenotype is a bit surprising because it is the telomere length acceleration, which is the aging phenotype. And we also observed significant strong associations with maternal smoking and intelligence phenotypes. I should also mention that among all the replicated associations, 79% are from the atypical POE class. This is quite unusual because the POE CPGs belonging to this group only accounted for 40% of the total POE CPGs. So that means a small proportion of POE CPGs explain the majority of the associations. And this observations was further confirmed by another analysis where we calculated the normalized associated CPG count for each phenotype. So we want to work out what is the phenotype that is associated with the largest number of POE CPGs. In order to work out that, we need to account for the local CPG correlation structure because, just like the LD structures in the SNP field, there are also local structures within the CPGs. So we only account for ones for each correlated CPG cluster. By doing that, we observed a much higher normalized associated CPG count in the atypical class, in particular for the smoking and telomerase acceleration phenotype. Since methylation association signals were detected in POE regions for some phenotypes, we next wondered that whether for those phenotypes there is an enrichment in terms of the methylation association signals in the POE regions as compared to the non-POE region. For this, we applied Mann-Whitney U-test to compare the significance levels of methylation associations from the POE regions as compared to that from the non-POE regions. The result indicated that for the atypical POE class, there is strong enrichment in the intelligence, aging, and maternal exposure, smoking exposure phenotypes. In particular, the strongest POE enrichment were detected in the verbal intelligence and the telomere length acceleration. In contrast, we didn't observe that enrichment in the typical class of the POE CPGs. We next hypothesized that some of the POE CPGs can function together in networks in the form of co-methylation modules. So we next identified the POE CPG-specific co-methylation modules in our sample. We performed the analysis independently using the discovery and replication cohorts, and the result showed that the modules identified in both cohorts are highly consistent, so that this allowed us to drive the matched modules, which include 6 modules for the atypical POEs and 8 modules for the typical POEs. For each module, we calculated the principal components based on the methylation levels of member CPGs for each module. This is to quantify the module activity, and we used the top pieces in the association tests with different phenotypes. As you can see here, the results indicated that the majority of the associations are contributed from the module identified for the atypical POE group. So this is quite consistent with what we observed at the single CPG association level. The most associated phenotype, again, comes from the aging phenotypes, smoking, maternal smoking exposures, and intelligence. When we characterize a methylation network, one way is to quantify the network activity, which can be done by the principal component analysis, which I just introduced. But there is another way to look at the network, which is to measure the network connectivity. So we next consider whether the aging-associated POE co-methylation modules displayed the stable or altered internal methylation module connectivity during the aging process. So following this idea, we identified a really interesting POE co-methylation module, the atypical POE module 3, for it displayed an increased internal methylation connectivity with age. we use a pairwise correlation of CPG members of that module as a measurement of connectivity. As you can tell from the left side of the figure, the pairwise correlation of that module increased gradually during the age. And you could also tell that when people getting older, the variation of the pairwise correlation also increased. So this suggested the existence of subgroups. And indeed, we performed subclustering analysis which identified subgroups, a flat group that didn't have the pairwise correlation increased, and a modestly increased, a sharply increased groups that have the correlation increase with the age. We next wondered that if there is any POE-CpGs that drive the increase of the connectivity of this module with age. So we look for the POE-CpGs that was connected with the largest number of other CpGs and tested whether those connections increased a lot when you compare the youngest and the oldest age groups. This allowed us to identify 5 hub CpGs that displayed these kind of features. Among them, the one highlighted with the red color displaying the strongest connections with other CpGs, and that connections increase a lot during the aging process. I should also note that 4 out of the 5 hub CpGs are located or nearby a gene encoding a protein interacting with the amyloid beta precursor protein, which is a very famous protein involved in the Alzheimer's disease pathology. So now we further explored a bit about the top hub POE-CpG for its association with aging, and surprisingly, we observed a dynamic association pattern with aging across different age groups for these CpG sites. As you can see that when we first look at the correlation between the methylation level of this CpG and the age, we observed a positive correlation between the two before the people getting old, middle stage. But after that, when people getting older, this correlation disappeared. However, when you look at the correlation between the methylation levels and the telomere length, we observed the opposite direction, where before the middle age, there is no significant correlation between the two. However, when people getting older, they become to be positively correlated with the strongest correlation detected in the oldest age group. And the same applied to the telomere length acceleration. When look at the genomic context of this top hub POE-CpG, we found that this CpG is in a promoter which was supported by the roadmap epigenome data and located in the downstream of a gene called CCDC115 and the upstream of a gene called IMP4. About the relationship between the methylation of this CpG and the expression of nearby genes, there is a recent publication that directly provide measured evidence that the blood methylation levels of this CpG is positively correlated with the expression of the IMP4. We also performed SMR analysis, which supported a genetically positive correlation between the brain methylation levels of this CpG and the brain expression of the IMP4. And we also find a database that provided evidence for the significant correlation of the methylation levels of this CpG in the brain and in the blood. So overall, this evidence supported that the methylation levels of this CpG can be indicative for the expression of IMP4. So the reason that we are interested in the IMP4's expression in the brain is because this gene encodes a component of telomerase which regulates the telomere length, and this protein has been found to be physically interacted with the amyloid precursor protein, which has been shown to be a landmark of the Alzheimer's pathology. And we think that the Alzheimer's disease is relevant because it is a very well-known aging-associated disease. So we extracted publicly available data for the brain expression and find that among all the brain regions available, only 2 of the brain regions displaying a significant differences between the IMP4's expression in the control group in the asymptomatic AD patient group and AD patient group. And these 2 regions are the temporal cortex and prefrontal cortex. The 2 brain regions are exactly the regions that are relevant to the Alzheimer's pathology. Finally, we also find that the atypical POE CPGs displayed some of the well-known aging-related epigenome features, such as high levels of methylation entropy, fast information loss with age. And we also find that the atypical POE CPGs have a high methylation correlation with clock CPGs, but we didn't see that for the typical POE CPG group. In conclusion, our phenome-wide association analysis at both the single CPG and the methylation network level identified strong and enriched associations between the atypical POE-influenced methylome and the human adulthood aging. A series of aging-specific analyses were further performed, which identified that one of the aging-associated POE co-methylation network increases the internal connectivity with age. We also observed other aging-associated features such as high levels of methylation heterogeneity, a faster information loss with age, and high correlation with clock CPGs in the atypical class of the POE CPG. So I hope all of these results could convince you that we have shown some POE evidence for the early development origin hypothesis for the adult aging. With this, I would like to thank many people's support from the University of Edinburgh and Sun-Yat Sen University to make these projects become possible. Most of the analysis were performed by my student Chen Hao, and I want to particularly thank the support from Paul, Chris, Andrew, because they have been supporting the POE project for a long time. Finally, thanks for the listening, and I'm happy to take your question.
There is one question on the Q&A that I can read out.
Okay.
So this is from Mike McGrew. If I understand correctly, there is a correlation of POE methylation with smoking, which also correlates with verbal skills and education. How do you eliminate socioeconomic influences from this analysis?
Okay, so thanks for the question. So first of all, for all the non-smoking phenotypes, we have accounted for the smoking effect in the model. So the smoking status and the pack years are the 2 covariates fitted in the model. And we also performed additional sensitivity analysis to use SIMD, which is a variable that is available in Generation Scotland, which describes the socioeconomic status. I think it's based on the region that people live in Scotland. And so it's kind of measure the socioeconomic status. So we perform analysis to see if we additionally account for that in our model, whether our associations results disappeared, but the result indicated that they remain still significant. So I think we have already ruled out the reported result, for the shared influenced by the socioeconomic status. I hope that's clear.